Okay, welcome to the next webinar in this series for LabChart Lightning. My name is Mackovski. I'm the Scientific Support Specialist with ADInstruments. Today we're going to go through a really exciting new feature within Lightning called CrossRecording Analysis, and that's to do specifically with the table. It's a really easy and efficient way to be able to collect the data you now analyze within your recordings or within your LabChart files into one simple data table. So you don't have to worry about always just copying something, then pasting it over to Excel as an example, or Google Sheets. This way, it's a really simple way to put your data together and do some extra calculations to really maximize what you can get out of your analysis. So let's go through that together. Okay, so what we have here is one of my Lightning files. So to do this, how to get the most out of the table here, what I've done is I'm doing an experiment, well, it's a mock experiment of just using our human non-invasive blood pressure device sold by ADInstruments. So this device itself, people just put it on their wrist, and we have a little cuff that's able to be placed over the fingers, or at least on two fingers, and you're able to actually measure multiple things here. So you can measure your systolic, diastolic pressure, which can calculate then your mean arterial pressure. You can also calculate your heart rate, and of course the finger pressure as well. So this is something that I am talking about, is just this here. So you can just see that we do sell this system here. It's really an easy and a non-invasive way to measure these parameters for people, and we are selling this right now at ADInstruments. So as we've calculated some data here, what I want to do is, for this particular experiment, I have recorded my baseline and several exercises. So what I want to do is compare how my finger pressure is, my heart rate, and my mean arterial pressure is relative to the exercises, compare that to the baseline. So what I'm going to show you here first is that this particular project I call it, the project is called the Bioreflex Sensitivity Project. And within this project, I have all these files. So if you remember from our last webinar, or if you have some experience using Lightning, that all these recordings here, these are all different LabChart files. So remember that a project is just the overall theme of what you're doing, and then just all your LabChart files are within this project. So for me, in this particular case, I have all these different recordings. If I click on any of these, you can see these go to different recordings. And this way, what I'm going to do now is collate regions within each one of these files to put it into a simple table to extract any data that I want. So the first thing I'm going to do is show you where the table is. Where is it? So it's right here in the show table view. And if I just click on here for the first time, if I don't have any regions selected, then you're just going to see this. Okay, so it's not like how in DataPad, when you go on DataPad in LabChart 8, you already see like an Excel sheet format. This is really totally differently designed than how we are with DataPad. So it takes a bit of learning, but once you pick it up, it's actually much easier to use than DataPad itself. So how we actually do collect data here is by using something called regions. We did touch on this on a previous webinar, but what a region is simply a selection. So for me to select my data, all I have to do is left click on a particular area. So if I want to highlight all of my channels, I have to go up on the time bar here and left click and move across. And you can see that that area there does a couple of things. One, it shows me my default means on the right here in the panel. The other thing it does, it gives me a little plus. So this plus tells me I can create a region. So with that region, then I can then actually start building my table up. Now in this particular case, I don't want to highlight all this. I just want to do specific signals. So how to do this is, now we're actually going back to one of our one of the original webinars on just moving signals around, and this is how we can do it. So first I'm just going to click escape, or click away from it. And what I'm going to do is, since I'm only measuring heart rate, mean arterial pressure, and finger pressure, I can just left click on my signal and drag it up to my finger pressure channel. I can also do the same thing with heart rate, drag it up and drop it in there. Now, look, I don't want to see any of this data. I just want to focus on this. So I can do this two ways. One way, I can just make this larger, but a much simpler way is just to use this option here, the maximize channel height. So by doing that, now I only have that channel. And by using my scroll wheel over my scale, I can change this. And of course, using the scroll wheel here, I can change my zooming out as well, all by using the mouse wheel, which you can do also with the trackpad on your computer. So as you can see in this particular experiment, we have some comments that are dropped here. You can see here. And what you can see is that I have some comments dropped for baseline, and then we have some for wall sit and wall start and end. We have one for static lunge start, static lunge end, and we have one for bicep curl, start and end. So what I want to do is create some regions. So now what I'm going to do is left click in an area like this, and I'm going to press plus. Now, I've actually created some regions already, but what I'm going to do is show you how to start with this. So what I'm going to do first, let me just remove these types of regions and start again. So then you don't actually see any of these. I was practicing this before, that's why you saw them. Okay, so now we have a region. If I double click on this, what you're going to see now is, let me just remove this and start again. If we highlight an area, press plus, like this, it shows me two options, either new type or no type. So with no type, that means there's going to be no, it's still going to be a region, but it's not going to be classified as anything. So you use the no type if you just want to look for a specific sample, but it doesn't mean more than that. You just want to look for a particular mean, or you can modify that, of course, to look for other calculations. But in this case, when we're doing the cross-file analysis, we want to use types of regions. So that's how we're going to be able to cross-file analyze other regions in other recordings by using the same region names. So here, we're going to call this baseline. So you can see up here on the right, we have a few options here. We have, once again, a little feature, just a little preview of the actual signal itself. And by using my scroll wheel, I can zoom in and out. I have the option to type it in, what region it is. In this case, I'm going to call this baseline. And if you notice here, there are these dots. Like I said in another webinar, that anytime you do see these three dots, that leads to something else. So it leads to some properties. So let's just investigate what these properties are. So in this case, if I choose on these, it shows edit baseline. So with this, I can, of course, change the baseline. I also have the option to change the color. So the color is actually similar to what we have with the comments, or the annotations, is instead of having blue, you can actually change this to any color you like. So for example, if I want to change this to yellow, I can do that. So that's handy, if you have a lot of different regions. If you're looking at a file, from if you really zoom out, it's really eye-catching when you have different colors. So it just helps you possibly just look for a particular region. So you can do that if you like. And of course, you have the option to delete region type, which I did a little bit earlier on. So if you have too many regions and you find one region is not really of use anymore, you can actually just remove that region as well. So if we go back here, and we also have another properties here. These properties here just tell us more about the start time, the start end, and the length of the region itself. So what I can actually do here is I can modify the length of my region. I can also change it by actually changing the value here as well. I can type in a value and it'll give me that. Another option here is a tag. So the tags are pretty common within Lightning, and this just helps you categorize your data. So in this particular case, if I notice something in this region that was of importance. So instead of me typing, like, for example, you can see just there was a bit of a space here when the NIBP was recalibrating. Instead of me just typing in baseline with a recalibration point in the finger pressure, saying something really long, I can just keep it as a baseline. But what I can do with the tag, I can just add something extra to it. So here I can just say, recalibration occurred, like that. And then I can just have that. And you can see instantaneously that's now become a tag. So what this does for me is that helps me search for these particular regions. So in this particular case, just if you wanted to know how to search, you can search for your recordings here. So obviously, as I've only done one region, if I just type in baseline, it will show me a couple of things here. If it shows you that symbol, that means annotation. This symbol here shows me a region. So if I click on this, it'll just directly show me that, okay, the baseline region is only in that signal, in that particular recording. Alternatively, if I had many baseline regions, in multiple recordings, instead of me just typing baseline, it's going to show me all these baselines and I don't know which one's which. I can type in the first letters recalibrate, and you can see there it automatically looks up that tag. And that tag is connected, of course, to that region, which shows you up here as well. So you have that option to see it there. The other way you can actually see it is in your regions view, which is a really powerful tool as well. So here you can do the same thing. You can look for recordings, so you can type in recording names, you can type in subject names, signals as well. So in this case, I'm looking at finger pressure, mean, etc. So if I type in finger pressure, it'll look for all the finger pressures, and it shows me I only have one finger pressure here. But of course, I can also look for that tag. And once again, that tag pops up, and I can search for that tag, and it'll take me directly to where that tag was in the region. So that's the handy tip when you are using regions view, and you can just close that to have that. So that's a bit of a tangent there, but that's what it means with the region. So if I double click on my region again, you can see it there, and I have my baseline. Other options here is you can actually remove your region by clicking on remove, and you have other options here of exporting. So what these options provide is I can either export as a text file all my data, or have the option of actually copying what this is. So copying is a really powerful tool, I find, especially when you are pasting something in Excel. So what you can actually do here is it will actually, I know for this particular experiment that it's calculating at about 25, well, it's sampling at 200 samples a second. So within that, every sample per second is actually going to be found within this region. So what I can actually do with this, I'll show you a bit later on in the webinar, but you can actually graph all of the data there into Excel or Google Sheets as an example, and that way you can just present regions, in your presentations. So that's a pretty handy thing to have. You don't have to just, do a screenshot. You can actually just collect all the data, put it together in a nice little graph, just to present to your colleagues or even for publication. So that's the baseline. Now I'm just going to keep moving on now to do some more regions. So I'm just going to move across here, and let's just choose static here. So we're going to press plus, and we're going to create this as a new region, a new type. So once again, I'm going to go static lunge, just like that, enter. So that's my second region. And I'll do the same thing with this one here. We're just going to left click here and make this a new region type, bicep curl. Enter, that's it. So now we've got 3 different regions. So let's look at that in the table, see what there is. If we go to the table view again, what we see now is if I just close this, you see a very simple table, but it's actually showing you quite a bit of data. All I've done was just drag and drop, and drag a few different regions, give them a name, and just because I put the signals, superimpose them on top of each other. So I remember I had the heart rate, I had the finger pressure, and the mean arterial pressure. I put them all in one channel. Now we have all this data. So it's a really simple way of just collecting a lot of data really fast. So what you can see here is a few things. So the top is going to be our regions. So you can see the baseline, bicep curl, static lunge. And within these, of course, they're going to be our different signals. So they're all the same because all of these are in the same channel. And what you see here is the mean and number one. So what do these mean here? So if I go back to, let me just go back to the channel here. Okay, so what you see with the chart view here, just FYI, if you ever want to go back to your chart view properties, just press the three dots here, and there's your table properties. So just like I said before, anytime you do see these three dots, it shows you some properties. And when you see this in the chart view, that's what we see in the chart view properties. So for this one particular experiment, this is human NIBP exercise number 1. There are no tags involved with this, but you can add some more tags if you wanted to. So like I said before, if this had some extra information involved with this particular subject, you can just add a tag here, So you can see that this person here is one subject. There's 6 channels in this particular recording, and you can always add some more channels here. And what we see here, that this recording is classified in a group as female. So we would assume that in this recording we have a female, and that's the group. And that what we can do with that by group, we can search groups, and B, more importantly, we can use the groups to help us categorize our table. So in this particular case, as we only got one recording and it's a female, you don't really see much here because it's all just standardized. But let me just add some more data to show you really the power of using the groups as well. So a bit more about this here. So what you can see first is the mean. So what does that mean? So basically the mean will just be the summary data, the summary calculation. So the first row will always be the summary calculation. So when I'm adding more data here, obviously here as n equals 1, it's just going to be the same values. But as I add more data, these values will obviously show my means. And I will show you a little bit later on how we can modify this to show not only means, but standard deviations, variances, maximum mins, counts, etc. So there are other options you can do to provide summary calculations, which is a bit more trickier to do in DataPad. You'd have to just do like a little Excel sort of calculation to do that. But here it's done automatically for you. And it's really easy and simple to put together. So let's add some more data here to make this table look a bit more convincing for you, just a bit more better understanding. So if we go back to our our chart view. Now let's go to the second exercise, the second recording. So this is pretty much the same sort of thing. What I'm going to do first is move my heart rate up here and my mean arterial pressure, like that. I'm just going to move this maximize. And just to show this, you don't have to maximize it if you don't want to. Like I said, you can just highlight it as is. So I can just highlight it like that. I can press plus. And now you see that any region that I made within this project, because it's in the same project, just we have all these different recordings, that these regions are saved. So now if I click on baseline, what happens now is that all that data will be under the baseline region within the table. And I now do the same thing with the lunge. I can press plus, static lunge, and bicep curl, like that. Bicep curl, great. Now let's just do one more as well before I move on, and we'll just move across here. And we'll do the same thing with baseline. Actually, before I do that, I need to move the data across. Baseline, static lunge. You can see how easy it is actually to do this. It's really simple to put your data together in a table. So just by doing a couple of clicks, now we go back to the table. What you can see now is I've added more recordings. So you can see number 2, number 3. These happen to also be females as well. So that's why it's all under the female category. If it was a male, it'd be on a different, it'd be a little bit further down here. It'd be kind of a separation between these two as well. But you can see here that these are my regions, and this is my summary data of these regions. Notice something that's really important is that when this table does get quite large, but you want to know exactly where that region is, something that's really nice to have in Lightning is the ability to navigate to that region. So anywhere here that I click, if I want to see, okay, I want to know something more about that, whereabouts in that chart is it, I can actually click on here to go navigate to that region, and it'll instantly navigate to that region for me. So that's a really handy tool to have, just to be able to navigate to wherever I want to go. It's really handy to have that. So that's how it works. So now what I'm going to do is finish these. I'm not going to do them all at the moment, as there are quite a few, but what I've done is I have finished this a little bit earlier on, and I'm going to go to the complete table version instead. So what we see now is the completed table. So with this table, now we see the female group. So this is when I was meaning that it's good to categorize what they are. So, if this is an animal experiment, we can see where ones with high fat, high sugar. One might be your control group, another one could be your sham group, another one could be treatment group, and you can just put them all together and then just categorize what sort of animal they are as the recording name, and then you can have a nice clean table like this. So once again, anywhere I click here will take me instantaneously to that region by clicking navigate to that region. So now let's talk more about the table itself. What can we actually do with this data? So what we see is the mean, which is great. That's really cool, but what if we want more calculations out of this? What do we do? The first thing we want to do is press these three dots here, show properties. By clicking on show properties, we are given these table property views. So what these will do is two things. Either A, it can show us more calculations, or B, you can actually hide some of your data. So even though the data here is nice and clean, you can imagine when I do talk a bit more about calculations, this table can get really full. And instead of, it can get quite confusing if you have too much data to present. And there are ways to hide data, not to delete it, but just to hide it. And you can export whatever you see. And that way it's a bit easier for you to extract data that way. So I'll show you how to do that. So let's just go through these options here. So the first option here is we can select region types. So right now we have all our regions selected. But let's just say, for example, I didn't want to look at static lunge at the moment. I just want to look at baseline and bicep curl. I can remove static lunge and it'll just remove it for me, just like that. Same thing with bicep curl. I can remove that and it just shows it like that. Otherwise, I can click on these and bring them back. So that's what this option shows. I'll talk about this a little bit later on, but let's just go through these two options here. Groups. So here I have male and female. If I just only want to look at the female, I could just obviously click here and remove that and have it that way. So that's something that's quite handy to have when you're having multiple groups together, but you only want to look at specifics. It's a really handy way of just categorizing your data, make it a bit more easy to see. And here's our summary calculations. So I will talk about this in just a second. Actually, I'll talk about it now. So what does this mean, summary calculations? So as I was mentioning to you before, up here in the first row, here and here, these are summary calculations. So these will be the mean by default, but you can add other options here. So let's just say I wanted to add a standard deviation. If I click on here, now you can see instantaneously standard deviations made for me. And of course, if I want to keep going on, variance, count, maximum, minimum, as examples, you can see how easy it is to add further calculations as your summary data to these. And this does take a bit longer in DataPad to do this, but you can see how quickly you can do this. And this will actually keep updating the more data you put in there. So it is tricky to do this in DataPad because you can do this, but then when you add some more data, then you have to modify the calculation. Same thing with Excel. You may have to modify that calculation as well. Here, it's just doing it for you instantaneously. So that's a really handy way to look at your data really quickly and to provide analysis. So that, once again, provides the summary data of all the data we have here. So that's what we have. So I'm just going to remove these for the moment. But now let's say we want to look at some calculations specific to these signals. What does that mean? So if we look at, for example, the finger pressure, if I click on this, this will show me all these options I have that I can add to my table. So right now I'm showing my mean, but let's say I wanted to add my median. Now I have my median there. Let's say I also want to add my timer maximum. I can add that. Or if I want, I can add my 1/3 max plus 2/3 min. Another option here we have is, all these options you can see. We can see our selection times and ends. We can have these points. So, and we also have options of comments as well. So in this case, were there any comments selected during this period of when I click to the region? I can see here, and I can see, well, look, I can see in this case that these comments were selected, or the annotations were selected within this region. So I can do that too. We have all these times. You can have time, of course. You can just plenty of different options you have for yourself. Block information I find is really important as well. So you can see you have integrals and slopes, which are of course very important with spirometry or with blood pressure, like PV loop as an example. Something here that I actually really enjoy is sampling rate. Some people may not know what sampling rate they're on, but here you can clearly see an easy way to show you that this particular signal was calculated or was recorded at 200 samples a second. We also have the same, something very important is samples and block. It tells us how many samples there are, and the block range as well. So you can clearly see the amount of information you can get with these other information here. So other options here, we have the cyclic measurements as well. So even though I have heart rate here, which was automatically created with the NIBP system, if I didn't have that, I can also calculate heart rate by using one of these measurements as well. So if I have this clicked, but if I choose an example, average cyclic rate, then this will be almost the same, but that is going to be your heart rate as well. So you can see how easy it is just to add some further data to my table. So you can see that the more data I click, it just keeps adding calculations, and it's a really simple way to do this. And look how, just by me clicking a few buttons, I have now all this data specifically just from finger pressure. So you can imagine that if you have a lot more regions, more signals collected, then having the ability to just be able to go here and then choosing what calculation you want is really convenient. So that's how this works here. It's fantastic little options we have with LabChart Lightning. Let's say, for example, we wanted to just look at some specific signals. So I'm interested here in only the mean arterial pressure. I don't want to see anything else. So there are going to be times when you're looking at a really large table, but you only want to see some specific signals. So how do I do that? So I did mention that you can hide regions and you can hide groups. You can also hide signals. All I got to do is right click and I can hide channel from table. I can do that too. And I can just do the same thing. Just keep right click, hide, hide, hide. And let's just say I was only interested in the females and not the males. If I just click on my properties again, I can choose to remove the male. And now I only have the heart rate for, well, the mean arterial pressure, my apologies, for females in this exercise here. So it's really handy to have. And all I got to do now, let's just say I want to export it. I can just go to export table view, and I'm going to put that on my desktop, save. And just as simply as that, by having that exported, there's my table. Really simple. So it's a really easy way to extract your data. So like I said to you earlier, that doesn't mean that the data is deleted. I can bring that back. Of course, I can just go back to fill out my tables. I can right click, show all channel regions like this, and do the same thing, export table view, save over. And you can see now the table has been repopulated with all my data. So it's a really handy way to work with your data here. Go back to our groups here. So now let's go through some more calculations. So another really important tool we have is the ability to do some further calculations, but with the summary data. So let's say for this instance, we're interested in looking at the differences between the summary or the mean between the bicep curl, the mean arterial pressure, versus the baseline. So how much of a difference is there? So what I'm going to do here is, once again, by pressing my 3 dots, I'm going to come up with my calculated channels. By clicking on here, I can go to create column. And by going create column, what this is going to do is going to only calculate based off the regions, but it's only going to look at the summary data. So the data here only on this row. Okay. So remember, this row is the summary of all my data here. So this can be your mean, it can be your standard deviation, etc. Like I mentioned before, you can provide different calculations here, but let's just stick to mean as it's the simplest. So what I'm going to do is I'm going to say, for this instance, bicep curl. And let's just look at the difference. So what you see here is a little place to do some basic arithmetic. So what I'm going to do first, I'm going to click on choose a region. And remember, this is only going to be using based off regions. We're not going to add any numbers in here. It's only going to use regions. So it's limited to what it can do at the moment, but in the future, hopefully we can add the ability to add numbers so you can work out things like percentage change, those sorts of things. So let's say, for example, we looked at bicep curl and then clicked on mean arterial. Okay, that's it right there. And now we're going to click on minus subtraction. And now we're going to go back to here and choose baseline mean arterial. So if I do that, you can see now it is calculated, the calculation is valid. And what you can see now is that it's giving me the difference with the female bicep curl. It's 23.38 millimeters of mercury, whereas the male is clearly larger. So that's pretty cool. So what else can we do here? So instead, let's just say we wanted to look at a factor change. So instead, now let's just do the same principle, mean arterial with a bicep curl, but divide that by the baseline. So now we can see the factor change here is 1.252 times bigger, whereas this one is clearly a larger factor change, which makes sense with male there as well. So you can see there, even with the average data among all of the subjects, we can see that there is a significant difference there, well, relatively compared to the females in this particular case. But this can be changed, so we can obviously change whatever unit we want, and here we can either add any data you want, subtract it, divide it. We have the options of also brackets, so you can add some sort of region calculations in brackets, and you can do some more calculations. And you can see the tool of this, it is quite important. Once again, you could do this in Excel, but it's really handy to have this. And once again, this will also update with the more recordings you have. So in this particular case, if I just once again, if I go back to my properties, to my summary calculations, but it says standard deviation. Now you can see the standard deviation is added to this. Same thing with maximum, minimum. And as I'm adding these calculations in the summary section, so are these calculations also being modified as well. So everything is being updated. With the more calculations you apply to these, then you'll see this as well. So it's a really handy tool to have. So that's basically how the table works and what you can do with it. So the next thing I wanted to show you was, we went back to here. Now, of course, this is under one subject. So that means that each recording has one subject, but let's just say, you see this option here, it has multiple subjects. So what does that mean? So I did cover this briefly within the first webinar. We'll go through that a little bit today. So we go back to File and choose one of my original projects. This is a drug project. So what this one basically is doing is I have multiple subjects here. So I have Ben, Mary, and James all under the same recording. And what you see in this colorful little chart here is that Ben is being measured with the BP, blood flow, and ECG. And Mary is also being done the same, and so is James. So what I've done here is I've been interested to see among these 4 recordings. So I have 4 recordings here, and you can see they're my 3 subjects. And within these recordings, I have done some different, we're looking at a drug like drug X. So we just want to see how that drug is affecting our subject's blood pressure. And what we see here is that I've added some tags to these recordings. So here, this one had no activity. These subjects had some low level, and the subject in week 3 had medium, and then in week 4, they had high activity. So we want to see how that drug is affecting their performance. So what I've done here is, once again, I've added some regions. So you can see here that in this particular case, I've added a baseline, and I've also added a treatment. You can see it there, baseline treatment, baseline treatment. It might be a bit easier to see it on a different actual recording here, baseline treatment, like that. So once again, I'm looking at what the treatment's going to do for these people. So these particular sets of people, Ben is categorized as my control, Mary is categorized as a drug treatment, and James is my sick group. So if I go now to my table, since I've actually collected data, with using these regions, what you can see now is a similar table to what we had before. But now what we're seeing is that these data that I've collected is over different weeks. So each one represents a different week because we have 4 different recordings. And in this case, James was only using the blood pressure 2 signal. James wasn't using the BP1 or BP signal, but whereas Mary was using the BP1, and as you can see, Ben was using that signal. So that's why we have these gaps. But what you can see here, once again, we have the mean calculations of our signals. And by having the ability to use multiple samples, what we have done here, Mary, James, and Ben, we can subjugate our data to show James subjects, but also the control, drug treatment, and sick groups as well. And once again, if we want, we can do some further calculations with these. We can do things like, as I was suggesting, we can do standard deviations if we want, something like that for our treatment. Same thing here, standard deviation, numbers of points, etc. RMS, 1/3 max plus 2/3 min to calculate the mean arterial pressure. And once again, all these options just to go through any calculation you want. So you can see how the table really expands really easily the more calculations we provide. So that's how the categorizing works with groups and having multiple subjects, which you can see here, just because I've clicked this. And that's about it. The last thing I did want to show you is just how you can quickly copy these regions over to your Excel sheet before we leave. So just going to go back to my completed table. And here what I'm going to do is, as I was mentioning, if I wanted to, let's just say, for example, I was interested in the static lunge and I wanted to present this at my next lab meeting. So what I can do here is, if I click on this, if I double click on this, it'll show me my region, and I have the option to copy. If I copy that, what I can do now is, if I go to Excel and then open up here, if I just simply press Control V and paste it in here, just give it a bit of a moment. Okay, so now that's actually showing me all of my sample points in that region. And now if I simply put these together, I can just go all the way down to the bottom, have all my sample points collected. If I go to Insert, my line, and choose a combo. So what I'm going to do here, because as I have two signals that are measuring in millimeters of mercury, but one is measuring in heart rate, I want to have 2 axes. So by going here to combo, what I can do is, let me just modify these to line. And I know that my last signal here is going to be on my secondary axis. So that's how my table is going to look. So just by going to Excel, now I have all my data. So that's my region right now. That's exactly what you see. So if I compare that to what I have here, you can clearly see now that that region here, this here, is just the same as this here. So it's a really handy way of just being able to copy your data, paste it in Excel, and just be able to show little regions like this, for your work. And then of course you can just add little comments, modify your axes and tables. It's a really simple way of doing it. So it's a really easy, convenient way to put this together for people to present their data. So that's all we have to talk about with cross-file analysis. You can have a lot of fun with this, just adding any sort of basic calculations here, adding some more summary calculations, being able to hide columns to fit them whatever you like. And of course, having the powerful ability to select regions and then to use the same region titles across multiple recordings to be able to put all the data together to be able to show a really easy table like this and just speed up the process of your analysis. So thank you very much for attending. If you have any questions, please ask me now. Otherwise, I'll make sure to send this out to everybody. And just before we leave, I also have the other option here to show you that to do with LabChart Lightning. If you go to our website, ADInstruments.com/products/LabChart/Lightning, you'll see the options here. Not only does it give you a 30-day free trial, but it also, by putting in some details, but gives you some stories on some of our customers and also shows you some differences between Lightning and from LabChart 8. So some things are still missing, but we are working very hard on this to develop with time. And of course, we'll be developing more modules and all the whole team's behind it right now. So it's a really exciting time to look into this. So just once again, just go to this website here. And please, if you have any questions, let me know. I can talk to my sales reps as well, and we can get back to you. So thank you very much for your time. And any comments at all, any questions? I'll just wait maybe 30 seconds to see if anyone has anything to ask. But of course, you can always email me, and we can even have a Zoom chat or if you like about this, and just go through any of these together and see if your data, if any of your LabChart files are, if this is something that's important, you think it could work. Let's just see if we can work on it together and see if it works out for you. So looks like there's no questions. That's fine. So thank you very much for your time, and have a good day. Thanks a lot. Bye-bye.